Pages. We've got integration or adaptation. It's called several different things depending on which paper you read. Expression, and then interference. In the integration stage, this is where typically they utilize Cas1 and Cas2 proteins, which are universal to most of the CRISPR systems, to read or take the foreign DNA and integrate it actually into the cell's genome there for referencing later on. These sections are typically called protospacers, and they are determined by their PAM sequences up in the front. You can see the little red thing over there was the PAM sequence. All right. Expression. This is when you actually have the viral or foreign DNA enter the cell, and then the Cas system activates, and you get the Cas9 transcriptions. One of the things to point out that's going to be important here is that in Cas9 systems, you have trans-encoded small RNAs called the tracer RNA, and that has to pair with the cRNA to form the guide to it. One of the things we're going to get into is that that's no longer the case with this new system. That pairing is cleaved by the RNase 3, and yeah, they're both required for the Cas9. Here we go. This is demonstrating it. We got Eugene here's the Cas9, and then his two guides, the tracer RNA and the cRNA. All right, so in interference, this is where it's actually cleaving it up. We get the Cas9 protein and the cRNA complex recognize the foreign regions, and typically they have the nuclease domains H and H in the RUF-C-like domain. RUF-C is an interesting protein. It resolves holiday junctions, which are these really cool, like four-way things that are in DNA. One thing to point out, though, is type 2 systems like Cas9 do utilize RUF-C and the H and H domains, whereas type 3 can have as many as 6 or 7 Cas proteins bound to the cRNAs. This new system is being classified as type 5, so it's going to be quite a bit different in its mechanism. Here we've got, Cas9 with his RUBC, his guide RNA, taking out all the viral foreign matter there.
So what was Eugene?
Yeah, that was Eugene and Abraham.
No, but it was Eugene.
Eugene is the Cas9, and Abraham is his guide RNA. Here we go. This is the CPF-1 system here. All right, the CPF-1 is a large, approximately 1,300 amino acid protein. It only utilizes cRNA. It does not require the tracer RNA compound or complex to form. One of the major differences between the CPF-1 and the Cas9 system is that this targets DNA that's T-rich protospacer adjacent motifs. That's particularly useful with like Plasmodium falciparum. Am I saying that right? Yeah, right. I mean, it's a very T-heavy organism, and this presents opportunities for us to do that. Another thing, sticky ends. The Cas9 system cleaves straight down the middle. We'll see later on as they elucidate their experiments here that we got a 4 to 5 nucleotide overhang on these. All right. They actually needed to determine the protospacer adjacent motif sequences for this new system. So they looked at how Cas9 was done. It was performed by Jiang et al. 2013. They used 1,024 PAM sequences to find the right one. I'm about to explain exactly what they did here in a second. The Cas9 PAM site was found to be random, and then two guanines there. And that's on the 3' end. Basically for this test what they did was they inserted the F. novicida protospacer locus into thousands of E. coli plasmids. And they used every single random combination that they possibly could on the 3' and the 5' end. And basically what happened was, if the CPF-1 system is going to work, it's not going to be able to uptake these plasmids, because these plasmids are going to have the correct PAM. So we're going to see here they actually constructed 2 plasmid libraries with 5' and 3', 7 base pair randomized sequences. They'll be interfered with by the CPF-1. Results indicate the T-rich PAM on the 5' end, and it's observed only in type 1 systems so far. And right here's basically what they did. The E. coli expressing the FN CPF-1 locus. And you look at the results here, you see that all of the ones that have heavy T's on them ended up being depleted. Whereas things that didn't have the correct PAM sites expressed no problem. The CPF-1 system did not interfere with it. This is some more elucidation of, or diagramming of the results. The middle T is set, especially, is a very prominent figure in making this happen. Let's see. All right, so we've got, they wanted to make sure that it was just cRNAs, or this is before they actually knew it was just cRNAs. For the cRNAs and the tracer RNAs, they they did a small shotgun, or small RNA sequencing. They found short cRNAs with 42 to 44 nucleotides in length, 19 nucleotides of direct repeat, 23 to 25 nucleotides of spacer sequence, and then no transcripts at all that are indicating tracer RNAs. And we're going to see that up here with the 42 to 44 repeating area. You see that it's reading less than 50 nucleotides. You get that real heavy there. And of course, the CPF-1 system does still utilize Cas1 and Cas2, which is universal for chopping stuff. It also does have Cas4, which is a little different from, it's similar to a type 3 system. But they're classifying this basically as its own system here. It's type 5. We're also going to see that they did this plasmid entry experiment once again, generating these mutants down here, where they've actually knocked out the Cas proteins. They've knocked out some essential stuff. We've got the CPF-1 full right here, which is plasmid with all the Cas genes and natural promoters. The CPF-1 min is plasmid with synthetic promoters, up the expression. And then the CRISPR array, and then a plasmid with all CRISPR genes. And then we do see here that, you have your empty vector. That expressed no problem. And then all 3 of these versions had significantly reduced expression, mainly because you don't need the tracers. You only need cRNA. Wrong way. They did a sequence on the cRNAs, and basically the Sanger sequencing, if anybody's familiar with that. Basically, you take your target RNA, or DNA, you throw in some specialized oligonucleotides that have one of the hydroxyl groups replaced with something that can no longer bind to the next carbon. So essentially, every time you throw in your polymerase there, every time it starts to build, it runs into one of these connections that prevents it from building any further. And you can just sit there, run your blot, and just read it from the bottom up. When they sequence this, it's really slow. Right, we got several different things here. The main thing, because they were trying to sequence the cleavage site here, and you see how you have the overhangs. It's not directly straight down one cut. That was the major finding here with this. They did find that the CPF-1 system can cleave linear and supercoil targets. And they found also with this that it's actually cleaving fairly far away from the seed region. The 18th base on the non-target strand and the 23rd base on the target strand. That's going to be very important because the Cas9 system cleaves very close to the seed region, which often disrupts it for further modification. With this, you can continuously use the CPF-1 system to edit the genome because it's far away from the necessary seed region. Where does the cleavage actually occur? We've got this also has the rough C-like domains, which again separates it from the Cas9.